what I'm going to talk to you about today are that there are such things as obesogens, and that these may contribute to this epidemic of obesity that we see in America and to some degree around the world. I'm going to show you a little bit of data because this is a public talk. It'll be less data than there would be in a scientific talk, but there's still going to be some. And I want to show you that obesogen action involves the reprogramming of the fate of a class of stem cells in the body to be predisposed to become fat cells. I'm going to show you some very new data that's not published yet that obesogen exposure modifies the response to diet. This is something that we've been trying to determine for a long time, and we found it, and we're very excited about that. And lastly, I'm going to show you that the effects of obesogen exposure are heritable and can be passed on to future generations, and that's the point of this picture here. This is 4 generations of a family. So by way of background, the observation is in the last 40, 50 years or so that non-communicable diseases are on the rise. That is, diseases that are not caused by infectious agents, bacteria, viruses, molds, things like that. And you could give a lot of examples. There's a few here. So leukemia and brain cancer have increased a lot since 1975. Asthma has doubled. People are having difficulty in conceiving and maintaining pregnancy, especially young women, and that's a worrisome fact. Autism has increased tenfold over the past 30 years. Why is that? There could be some amount of increased diagnosis, but there's a real phenomenon happening here. Why is that? And obesity has increased more than 3-fold in the US over the past 40 years, and it's doubled worldwide. So one of the things my laboratory is interested in are so-called gene-environment interactions. So how does our genetic makeup interact with factors in the environment? The important fact is there's no way possible that our genetic makeup has changed in 40 years. It's simply off the table. It hasn't happened. There's a lot of, we can call them quote-unquote epidemics of chronic diseases, diabetes, childhood asthma, ADHD, obesity. There are lots of them. These cannot have genetic causes. These are caused by something else. And I would say, how can we understand the etiology, the origin of these diseases, independently of considering the environment? The NIH is focused on genes, and they largely ignore the environment. So here's the NIH model. Why do certain people develop diseases, why other people don't? That those certain people have different genetic susceptibilities, and that leads them to have adverse health outcomes. And there is acknowledged that poor nutrition can play a fact, and that if you have one disease, it may make you more susceptible to another, and that stress and drug use, and there's a whole variety of what are called comorbidity factors. And there's one tiny little institute at NIH, the National Institute of Environmental Health Sciences, and they study the role of environmental factors in the etiology of disease. So I'd like to introduce something that I only became aware of about 10 years ago, and that's this idea of the developmental origins of health and disease. And this derives from the work of a British physician named David Barker, who observed that in the north of England, in Wales, that people were dying early of heart disease in their 50s. And he could trace, using birth records, that many of those people who were dying young were born small for gestational age. They were born as small babies and they died young. And that led him to postulate that maybe this reduced growth during fetal life is associated with chronic diseases later in life. And you can see this has even made it into the popular press. This was in Time magazine a few years ago. His colleague, Mark Hansen, generalized this idea of developmental origins to what's called the Dohan hypothesis. And the whole idea is that the fetus adapts to conditions that it encounters in the womb, and those adaptations are permanent. So if the fetus is encountering very poor nutrient status, it changes metabolism to utilize those nutrients effectively. And if nutrients are not scarce later in life, that will cause a problem. There's another very famous study that was done, you may have seen the Nova about this, called The Ghost in Our Genes. Studies in a very northern place in Sweden called Uverkalix, where it was shown that the food availability during a child's life between 10 and 12 years affected the longevity of his grandchildren. Think about that one. And surprisingly, if food was relatively scarce during that time, his grandchildren lived longer. And if food was abundant, the grandchildren lived less long. That illustrates the possibility that this programming of cell number and metabolism continues into adolescence. This also made it into Time magazine. And we know from studies done in many laboratories around the world that there are diseases that have developmental origins. That is, diseases that are affected by the conditions that a fetus or an embryo faces early in life. Cardiovascular disease, there's a whole list of them. And I'm going to talk to you about obesity and diabetes and how these can be influenced by early life factors. Also, I want to mention that this developmental programming is almost totally epigenetic. So what is epigenetic? That may be a word that you've heard or haven't heard. So epi literally means on top of. So epigenetic means on top of genetics. And it was coined by the famous embryologist Conrad Howell Waddington. And Waddington had the idea that early in development, you can remember we all start as a single fertilized egg, and that the cells have many potentials. And as development proceeds, there'll be an influence that acts on this cell, an embryonic induction, and it will divert it to one pathway. Then another time, a factor may happen that will divert it to another pathway. Once these cells have made it to their final fate, it's hard to change them again. So it's hard to change a heart cell into a liver cell, for example. So once cell fate is specified, it's stable. Maybe a more modern analogy would be a pinball machine for, I see enough people here that are old enough to remember what are pinball machines, where you start the cell, you shoot it off on its fate, and it interacts with various factors, some chromatin modifying factors, histone deacetylases, and it rattles around being acted on in different ways until it ends up in one place or another. The important thing to remember about epigenetics is it involves changes in the expression of genes without changing the sequence of the DNA. So there are no mutations. We're used to thinking of genetics. Something happens, radiation happens, causes a mutation, and that mutation leads to a mutant. Epigenetics involves no mutations. It's heritable and it's maintained once it happens. Epigenetic changes are reversible, although we don't know how to do it. We don't know how to specifically reverse these changes, but we know that in principle they're reversible. It's encoded in chromatin, which is the structure of DNA and its associated proteins, and it's a form of cellular memory. There are many, many examples that you're all familiar with. All the women in this room have two X chromosomes. One of those chromosomes is shriveled up into something called a bar body. It's inactive. Genes are not transcribed. That is an epigenetic modification. The genes on that chromosome are silenced. There's a wide variety of genes in cancers that are turned on or turned off by epigenetic modifications. Think about that. So if you mutate a gene so that it doesn't work, that's the classic mutation. But if you cause an epigenetic modification so that the gene's not expressed, same thing, right? Doesn't work, not expressed, the same thing. You don't have a protein, you don't have a function. So something happens that shouldn't happen, or doesn't happen that should happen. This is epigenetics. So you're familiar with diseases you learned about in school. There are some classic genetic diseases. Sickle cell anemia, caused by a single mutation in hemoglobin. Cystic fibrosis is caused by a single mutation in a chloride transporter. There's a whole host of these. Huntington's disease is caused by increasing the number of repeated sequences in a gene. There are lots of genes that have an epigenetic component, and we know this from identical twin studies. So here is a chart, and you can see that identical twins are in white, and fraternal twins are in gray. And if you look at various traits and diseases and mutations, they're not so closely associated. So look in height. Identical twins are almost always close to the same height, fraternal twins less so. If you look at the prevalence of Alzheimer's diseases, you're getting close to the 50% point. One twin has it and the other doesn't. Right? These guys are genetically identical. If you look down here to breast cancer, there's almost no correlation. One twin can have it, and there's no relationship with the other twin has it. So that means the environment is playing a role here. It's not just the action of genes. It's the interaction between genes and the environment. So that leads me to our story, the worldwide obesity epidemic. And 35% of the US population is clinically obese. That is, has a body mass index higher than 30 meters per kilogram, meters squared per kilogram. That's twice the worldwide average, and it's 10 times the average in Japan and Korea. And I gave this talk in Korea a few years ago, and they very proudly said, you know, only 3% of Koreans are obese, therefore Korean food is very good. It's hard to argue with that. You know, 68% of us are overweight, and that's estimated to come close to 100%. When I first started giving this talk, 2020 seemed pretty far away, and it's not so far away anymore, is it? Let me talk about BMI. So here's some images of two obese Swedish men studied by my friend Lars Lind, who studies old Swedish people at Uppsala. This is an MRI image, and in gray is fat. Right? So all of this around here is fat. So this person has rolls and rolls and rolls of fat. Looks terrible. But this person is metabolically healthy. There's no other health problems aside from being fat. This person is carrying all of his fat inside the abdominal cavity. That's so-called visceral obesity. This is what's associated with all of the adverse health consequences of obesity. And this person has had stroke, he's had a heart attack, and he's diabetic. And here's another person with exactly the same body mass index, our former governor. And you can say many things about this guy, but you can't call him obese, right? But 3 body mass indexes, exactly the same, totally different phenotypes. So you need to be careful. This is a kind of a blunt instrument that we're using to measure obesity. These are some graphs from the CDC showing what's happened with obesity in the US. You can see that in 1990, not too many of us were obese. Relatively small number. And in 2008, and there's a reason I showed 2008, many more of us were obese. So this other number has doubled over the same period of time. And I always offer the beverage of your choice to anyone who can guess what this number represents. It represents a real number. You don't count, Dave, because you've heard this before. This represents the number of health clubs adjusted for population. It's also doubled at the same time as the obesity epidemic has doubled. So you could cynically say that health clubs cause obesity. It's probably not true, but you know, that's what the data could be interpreted to mean. Here's a graph from Europe, and you can see that the color code is not the same, but you can see that the distribution of obesity over Europe is not uniform. There's some places like Scandinavia, for example, people are not so obese, but in Sweden, they're very concerned that the rate of morbid obesity has increased 5-fold over this time, from 1% to 5%. You can see here that, unfortunately for them, the Greeks are the most obese people in Europe. And you know, Greeks have lots of problems. And I saw this T-shirt in the Plaka in Athens a few years ago, and it's also impairing their ability to reproduce. The poor Greeks are in big trouble. Seriously, the reason that we're concerned, or one of the main reasons we're concerned about obesity, is because it adds a tremendous burden to the healthcare system. So the last number that I heard is something like $210 billion in 2008 dollars. That's serious money, even by Washington standards. And whenever I talk at a UC, I joke how many more vice chancellors we could afford with $200 billion. That cost comes from the association of obesity with increases in metabolic syndrome, which lead to type 2 diabetes, to cardiovascular disease, hypertension, stroke, also to cancer, to lost days from work. There are many, many costs of obesity that feed into the healthcare system. So it would be good if we didn't have to have those costs. If people weren't obese and the healthcare system didn't have to pay for these added complications. So how does obesity occur? And the prevailing wisdom that you'll hear from the vast majority of medical professionals is that it's caused by positive energy balance, as I sarcastically call couch potato syndrome. That means you eat too much and you exercise too little. And here's a classic example from the Disney movie WALL-E. And remember, these people were pampered and shoveled around for 700 years and they ended up like this. So this is the model. But remember what I showed you about health clubs. And we know that there are lots of other factors. It's not just strict calories in, calories out. We know that stress and increase in the level of stress hormones like glucocorticoids is associated with obesity. How many students here have been stressed out and gone for the Ben & Jerry's? Or whatever is your local favorite. We know that too little sleep causes obesity. And we know now that that's by a disruption of the circadian rhythms, your body's natural rhythms. We know that there's such things as thrifty genes that evolved during the days when we were hunter-gatherers to enable us to use calories well. So if you have to go out and dig up your food, or if you have to kill an animal and bring it back, the ability to store more of those calories is a selective advantage. You're going to do better in times of famine than someone else. And we hope that never becomes a selective advantage again, because that means civilization will have fallen. We know that there are things like adeno-associated virus 36. We know that the kind of bacteria that live in your intestines, the ratio of Bacteroidetes to Firmicutes bacteria, predicts whether you'll be obese or not. And we can take gut bacteria from obese mice and put them into skinny mice and make those skinny mice fat, just by changing the bacteria. Not by changing genes, not by changing diets, just by changing the gut bacteria. And we can also do the opposite. We can take bacteria from skinny mice and put them in obese mice and make those mice thin. So there are many more things than just calories in, calories out. We know from the studies in Southampton and also from the famous Dutch famine studies that too little or too much calories during pregnancy makes a child predisposed to obesity. Mechanisms are different, but the effect is the same. And we know one of the best epidemiological correlations that there is that maternal smoking during pregnancy leads to decreased birth weight and increased risk of obesity later in life. There's now 35 studies that are 100% in agreement. It's not often that we get that kind of agreement in epidemiology studies. So is there a role for industrial chemicals? So there was a woman named Paula Bailey Hamilton who published a paper. It wasn't such a great paper, and it wasn't in such a good journal in 2002. And she said that the reason that she couldn't lose weight after having her 4 kids was because she was full of chemicals. And she showed a graph that correlated the increased use in chemicals since World War II with obesity. And yeah, you can make lots of those graphs. I showed you the data for a graph with health clubs. You could make the same graph with SUVs, with global warming. There's lots of correlations. But we need to find causation. And Jerry Heindel at NIEHS published a paper that really got a lot of our attention, and he asked the question, could there possibly be a link between endocrine disruptors and the obesity epidemic? Because although Bailey Hamilton's idea wasn't really correct, that it isn't just chemicals that you can detoxify, she made a very important observation. And that was that there are chemicals, some chemicals that at high doses cause animals to lose weight, but at low doses cause animals to gain weight. So that was the really important thing that came from her paper. So Jerry wrote his paper, and some of us listened. And the reason that this is important is because there's some chemicals that have effects on the endocrine system, as Dave mentioned. You probably know that weight in your body is under the control of hormones. The most famous of these is probably leptin, which is the energy balance hormone. There are others. An example of one that we work on is the thyroid hormone. So that's a hormone that controls your basal metabolic rate. It controls how many calories your body needs to run itself. The development of fat in fat cells is also under control of hormones. So they're largely regulated through what are called nuclear hormone receptors. So these are receptors that live in the nucleus that respond to hormonal signaling. And the key regulator has the terrible name peroxisome proliferator activated receptor gamma, which is an artifact of history why it's called that. But basically it's a fatty acid receptor. And this receptor is the key regulator of fat cell development. If you express it in a stem cell, that stem cell is now a pre-fat cell. If you activate it in that pre-fat cell, it differentiates into a fat cell. And if you activate it in a fat cell, that cell accumulates lipid from the environment. So this is a very important player in obesity. So let me also introduce endocrine disrupting chemicals. So this is a little bit controversial. There's a lot of definitions for endocrine disruptors, so I'm going to use the definition that comes from the Endocrine Society. And the Endocrine Society says that an endocrine disruptor is an exogenous, from outside the body, chemical or mixture of chemicals that interferes with any aspect of hormone action. And that differs from the definition that the EPA uses, because the EPA would add, and causes adverse consequences. And there's a lot of debate about that. To an endocrinologist, inappropriately activating a hormone receptor is per se an adverse consequence. You don't need any more evidence than that. But to old-fashioned toxicologists, not like Dave and Ken, they need more evidence than that. So that can be the wrong signal, the loss of the signal, or the signal in the wrong place at the wrong time. And the key feature is these hormones work at low concentrations, and so do endocrine disruptors. So for example, the sex steroid hormones, estrogen and testosterone, work at less than 1 part per billion. That's the level in your body at which they normally act. So it doesn't take much to disrupt that. 1 part per billion is very, very little. That's a few molecules in a swimming pool. So how are we exposed to these? We're exposed to these are persistent organic pollutants, they're components in the diet, personal care products. There's lots of ways. We actually live in a sea of endocrine disruptors. This was something that in the US wasn't really widely accepted for a long time. And in 2009, this paper came out from the Endocrine Society that laid out the evidence for the existence of endocrine disruptors and the adverse consequences that they might have. And they concluded that the evidence for adverse reproductive outcomes is very strong, actually beyond dispute. And that there's mounting evidence for effects on thyroid, neuroendocrine, obesity, metabolism, and a variety of other things. So they had a statement asking for more research on this, and it led to something called the Endocrine Disruptor Protection Act of 2009, which sadly didn't go anywhere, which would have moved research away from the EPA, which is very influenced by industry, to NIEHS, which is not. The American Medical Association adopted a resolution calling for new policies to decrease endocrine disruptor exposure. And in Europe, the European Parliament voted a couple of years ago to direct the European Commission to present laws on the ban or the restriction of endocrine disruptors by 2015. And they didn't. Why? Because the industry influenced them. So actually, Sweden sued the European Commission and said, you must do this, it's the law. So that's very contentious in Europe right now. Here's a statement that I don't like. It says, the relationship of human diseases of the endocrine system and exposure to environmental chemicals is poorly understood and scientifically controversial. By Bob Kavlock, who's the director of the endocrine disruptor program at EPA. Thankfully, he retired. So perhaps there's some hope. His replacement seems more reasonable. So how are we exposed to these chemicals? This is a list that comes from my friend Jerry Heindel at NIEHS. And you can see that there are many chemicals. It's a big list. And there's some famous ones here you've heard about. Pesticides like DDT. You've heard about DDT. You've probably heard about atrazine. There's a whole bunch of others. These are color-coded by the pathways they act on, which isn't really important for this audience. An important point is these chemicals, herbicides, fungicides, and insecticides, are designed to be toxic to life. Right? They're designed to kill living things in one form or another. So perhaps it's not surprising that they have these effects on the endocrine system. But it's a very big surprise that all these industrial chemicals, which are not intended to be toxic to living things, are also endocrine disruptors. You've heard bisphenol A, there's been a lot of noise about that. There's a whole bunch of them. So we're exposed to these in many, many ways. Pesticides, herbicides, flame retardants is a big issue. Surface protectors. There's over 1,000 known endocrine disruptors. And it's surprising and it's also concerning because there has been no systematic effort to identify endocrine disruptors. These were discovered by accident, by people studying, like me, studying something else, and they found that, oh, this chemical affects this endocrine receptor. So I don't know how many there would be if we ever mounted a systematic search of the more than 80,000 chemicals in commerce to see how many of these are endocrine disruptors. We don't know. Sadly, we willingly expose ourselves to endocrine disruptors all the time. Personal care products are chock full of endocrine disruptors. Parabens, benzophenones, avobenzone, phthalates, they're everywhere. And if you believe, like many people do, that, well, they're there, but they don't really get into our bodies very much. If I put on a lotion that has phthalates, is that really a problem? And these two Canadian guys wrote a book with a very silly title, Slow Death by Rubber Duck, but it's about a very serious topic. So what these guys did is they used themselves as guinea pigs. So they had blood levels of a variety of chemicals measured, and then they sat in a room and sprayed an air freshener and watched television, and then had the levels measured again. And they sprayed Scotch Guard on the couch, and they sat there, and they measured the levels of chemicals. So just by living their normal lives and interacting with chemicals in the way that we do all the time, these got into the bodies. So exposure is there. The counter-argument is these guys from industry, and they would say, we in the business community prefer a cautious wait-and-see approach over needless media scaremongering. And this is, of course, the issue. That's what they say, that this is media scaremongering. And it's interesting because this strategy came from here, from the tobacco industry, who made the game plan for how to argue against all kinds of scientific effects. And it's also very funny that this guy has grown very large breasts, and he hasn't noticed that there are estrogens in the environment that might have played some role in that. So this is one point of view. I can argue against that with some data from Tracy Woodruff at UC San Francisco. This is a table from a paper of hers showing that a variety of disorders of reproduction and other hormonally sensitive things is on the rise in the past 50 years. And that reproductive function is on the decline. Sperm count is on the decline. Fertility is on the decline. Many factors that are associated with hormones being affected. So perhaps it's a coincidence that we're putting hormonally active chemicals in the environment and we have effects on hormonally sensitive processes. That could be a coincidence. Or maybe it's not a coincidence. The evidence is out there. Of course, this is the point of view of industry. They have their head in the sand. And we hear things from these guys that say, well, are endocrine disruptor effects really adverse? Is it a bad thing to activate a hormone receptor? And a group of toxicology editors got together and said that non-monotonic, means nonlinear, low-dose effects defy common sense and must be wrong. And at the Toxicology Society meeting last year, where Dave first invited me to come here, they had a public forum and they decided that they had a vote about whether there were such things about low-dose effects after a debate. It was a very silly thing. The data exist. The most silly thing that anyone says is, how can we believe animal studies, right? So the companies do studies in animals and say, our studies, these chemicals are safe. But Blumberg, your studies were done in animals, therefore they're not valid. And I say, pick a position. I don't care which position it is. So if you believe that effects in animals predict effects on humans, then if their safety effects are to be believed, then so are our adverse effects. And if you say effects in animals do not predict effects on humans, then you can't say that the chemicals are safe based on their experiments. You have to be consistent one way or the other. And they also say that effects are transient and will disappear after exposure stops. And we'll address that later in the talk, but the short answer is that's incorrect. Okay, so enough about endocrine disruptors, and we'll get back to provocative things at the end. Here's what we're studying. So is it the case that endocrine disrupting chemicals are leading our kids and adults to look like this? Is there some relationship between disturbances in endocrine signaling and this outcome that we don't want to have? That's a silly cartoon. Sadly, this is reality. There are lots of fat American kids. And the problem is that kids that look like this very often lead to adults that look like this. And I'm constantly dodging these little carts at Costco. You see them proliferating everywhere. It's probably not a good idea to solve this problem by doing bariatric surgery on people like this. It will help them, but that's an even bigger cost to the healthcare system. It's probably a better idea to help people not become obese in the first place. So that's where we hope the work that we're doing will lead. So Dave mentioned the word obesogen. So according to me, obesogens are chemicals that inappropriately stimulate the development of fat cells and storage of fat into those cells. And they can do that directly by functioning on the fat cells, Or they can do that indirectly by affecting appetite and satiety, so hunger and feeling full, or they can affect metabolism. So is there any evidence to support this, or is this just my way as a scientist to scam you all out of money to support my efforts? Well, we know from the work of Rita Newbold at the National Institute of Environmental Health Sciences that if you give mice in the first 5 days after birth a chemical called diethylstilbestrol, which is synthetic estrogen, which doctors used to prescribe to women during pregnancy in the 50s, if you give them 5 parts per billion, think about how little that is, for 5 days after birth, and then never again, you got mice that were really fat like that. Interestingly, if you give them 5 parts per million, 1,000 times more, they get skinnier. So that's an example of a dose-response curve that a traditional dose makes the poison toxicologist says, I don't believe it. The data are there, but it's not the way it's supposed to work, therefore I don't believe it. Rita showed that the soy phytoestrogen genistein can do similar things, and a variety of people have shown that bisphenol A, this chemical in plastics, can also do similar things. We've done the experiment in humans. We know that if we give them drugs that are called diazolidinediones, the name you may have heard is Actos or Avandia, which are diabetes drugs that are now black labeled. But if you give these drugs to people at any age, they get fat. It makes them insulin sensitive, which is a good thing, but it makes them fat, which makes them insulin insensitive, which is a bad thing. So this chemical, which activates this hormone receptor, PPR gamma, makes people fat. We know that phthalate levels in urine correlate, there's that word again, correlate, with waist diameter and insulin resistance in humans. From NHANES studies, the National Health and Nutrition Study that CDC does. And we know now that about 20 or 30 chemicals can be found in epidemiological studies associated with obesity. So that is, if you measure levels in obese people and non-obese people living in the same area, that these chemicals are enriched in obese people even after you correct for body mass. So that doesn't prove that they cause obesity, but it says these chemicals may be involved. Why don't you go test it in the lab and see if you can make that link? And we know that there's a bunch of chemicals, probably 50 now, that we can show in cell culture cause cells to become fat cells. So I would say that the existence of obesogens is plausible, that when we started these studies, there was already enough evidence to say that this is probably something worthwhile. And now the evidence has grown quite large. So here's our favorite endocrine disruptor, tributyltin. And tributyltin is an example of a chemical for which there was almost no controversy. So at parts per billion in the environment, it's found at parts per billion, and at parts per billion it causes this thing in snails called imposex. So snails are hermaphrodites, they have male and female parts. And if snails are exposed to tributyltin, this is the penis here, and look what happens to it. It gets colossally big, 100 times bigger. And as you can easily imagine, the parts don't fit anymore and these animals can't mate. So they become sterile. And that's the reason tributyltin is banned on ship hulls where it used to be used. I became interested when I was sitting very sleepily in a meeting in the south of Japan. It was all in Japanese and I was jet lagged, so I was hardly paying attention. And Professor Tanabe from Ehime University stood up and he said tributyltin could sex reverse genetically female flounder. You could make animals that would be 100% female, make 30% of them into males. And I said, what? Sex reverse flounder? Now you got my attention. So I called back to the lab and I said, guys, test tributyltin on all 48 hormone receptors and tell me which ones it activates. Because we thought maybe it activated a steroid receptor. Because the way that you make body parts different, the ways that you grow a penis on a female, is to affect steroid hormones. And what we found was that tributyltin bound to and activated at parts per billion, again, very tiny number, RXR and PPR gamma, which work as a heterodimer to regulate adipogenesis. So when we made that finding, there was only one way we could go with this. I never was interested in obesity before that moment. We worked on other things. But there was either ignore the result or follow it. So that's what we did. This was 2003. We showed that TBT could induce cells in culture to become fat cells. And we showed that if we gave it to mice, they got fat. We gave it to mice in utero. So I won't show you much of that data. It's published a long time ago. I'll show you one figure from that first paper. So this is the testicular fat pad, the fat around the male gonads. And you can see that in tributyl-10 exposed animals, it's about 15% bigger. Importantly, these animals were on a normal diet. We didn't give them a high-fat diet. They weren't eating Big Macs. They were eating normal mouse chow, but yet that fat pad got bigger. Second point is, the animals didn't get bigger. So they weighed the same, but they had more fat. So that means they made more fat at the expense of something else. And we'll come back to that. So we've been asking the question in recent years, how does this TBT exposure cause weight gain? What are the mechanisms? You know, we're molecular biologists. We want to understand what are the mechanisms through which this happens. So the first, most obvious one is that, well, maybe the animals eat more, or that they never know when to stop eating. There's a human syndrome called Prader-Willi syndrome, where kids never know that they're full, and they keep on eating and eating and eating. Well, the fact is that the animals don't eat more. They actually eat a bit less. Okay, strike one. Maybe we've altered the ability of the fat cells to store fat. Maybe we've made them into super fat cells, so they do an extra good job of storing fat. There's not much evidence to support that. The model we favor is that we've made more fat cells, or more pre-fat cells. The reason we favor that model is because when we started these studies, it was already known that there's a class of cells in the body called mesenchymal stem cells, which are found in many places in your body, and these cells are responsible for regenerating tissues in your body. So it used to be thought, for example, that you're born with all the cells you're ever going to have. You're born with all the fat cells you'll ever have. That's not true. Fat cells live for 10 years, and they're replenished from mesenchymal stem cells. Bone cells are replenished from mesenchymal stem cells. Same with cartilage. Actually, we're now using these cells to make new bone and cartilage in humans. So these cells were known to differentiate into fat cells when you give them this chemical called rosiglitazone, Avandia. So we made the hypothesis that perhaps tributyl-10 is causing these cells to become fat cells when they shouldn't be. So here's again the mesenchymal stem cell. It can make muscle, and it can make cartilage, and it can make fat, and it can make bone, and a few other kinds of tissues. And a key point here, this is complicated with lots of genes and stuff. Look right here. Here's our friend PPAR gamma, and PPAR gamma expression determines whether a cell becomes a fat cell or a bone cell. So remember, that's the key player that we talked about. So we asked the question, if we give mice tributyl-10 when they're pregnant, or we give them rosiglitazone, or we give them just vehicle control, and then 8 weeks later, isolate these cells and ask the question, have we changed them in any way? So we did that, and we could show that in cells which came from animals that were exposed to the vehicle, just the solvent alone, you got about 20% fat cells. And then cells that came from animals exposed to rosiglitazone or tributyl-10, you made about twice as many fat cells, or cells that looked like fat cells. So we looked at, we're molecular biologists, so we look at molecules. This gene, fatty acid binding protein 4, and here's leptin, which you've heard about, are also increased. So the cells look like fat cells, and they express the genes that fat cells express, probably the fat cells. At the same time, if we tell these cells, you're going to become bone cells by giving them a set of chemicals that make them do that, we found that the ones that came from the animals exposed to the vehicle made about 80% bone cells. And if they were exposed to rosiglitazone or tributyl-10, many, many less made bone cells. And that's reflected by markers like osteopontin, osteocalcin. There's a whole host of genes that are expressed in bone cells. They're all downregulated. At the same time, here's a fat marker, fatty acid binding protein 4. So these cells were instructed to become bone cells, but instead they became fat cells. So this exposure reprogrammed the fate of these cells to want to become fat cells no matter what treatment they were given. So we next asked, are these effects heritable? And in order to do that, we changed the experimental design a little bit. So in this experiment, we gave them 1 dose of tributyl-10 at day 14 of pregnancy. So here what we did is we gave them the same dose of tributyl but throughout pregnancy, starting from before pregnancy until birth. So they got it in the drinking water, so they were exposed throughout early development. And the reason that we did that is because at about day 9 of mouse development is when you can affect the fate of the germ cells, the eggs and sperm. So in order to see whether something was heritable, we had to expose them during that period of time. So we gave them different treatments. We gave them vehicle, we gave them rosiglitazone, and 3 different doses of tributyl. And this number here is important, the no observed adverse effect level. That's a toxicological term. So whenever a chemical is tested, and the few times that it is, the company that makes it gives it to animals at different doses, and they find out, this dose kills all the animals, this dose only kills half, this dose has just a little bit effect, that's called the low L, the lowest observed adverse effect level. Then they determine this dose called the no L, the no observed effect level, and that's the dose that's used to set human exposure. So the rodent no L, they apply a safety factor, usually of 100, to that and say, okay, that's the human exposure. So no one can be exposed to more than that, and then they never bother to test. So we're testing 50 times below that level, 5 times below, or 2 times higher. So what we did is these moms were exposed during pregnancy, and here's the embryos inside, and we took, the babies are called F1, and we killed some and we mated some, and their babies are called F2. We killed some and we mated some, and their babies are called F3. And we looked at a variety of parameters. We looked at the body weight. We looked at how much fat they had. We looked at the size of the fat cells. We looked at what genes were expressed in the mesenchymal stem cells. And we looked at the liver. If you see an effect in F1 and F2, that's different from an effect in F3. So the F1 and F2 is called a multi-generational effect. And that's still plenty bad. But this is different because in F1 and F2, the animals are exposed to the chemical. So the F1 is exposed as an embryo, and the F2 is exposed as the egg or sperm inside those embryos. F3 have never been exposed. So this is called a transgenerational effect. And my colleague on the other side of the state, Mike Skinner, is the one who's done a lot of this work and who makes these definitions. So if you see an effect in F3, the important thing is this effect is going to last. So Mike is going all the way out to F6. So think about that. So if F3 is your great-grandchildren, what is F6? That's a whole lot of years of your family. So what did we find? So we looked at the size of the different blobs of fat. We call them fat depots, because mice don't get fat everywhere the way we do. They collect the fat in certain places. We could see around the testis, the epididymal, that there wasn't so much there. Around the kidney or between the back, the interscapular. In the F1, we could see a small effect on fat cells. But in F2 and F3, we could see a big effect. Okay, so in all 3 generations, we caused increased size of the fat depots. If we look at the size of the cells, that was very interesting. In all generations, the fat cells were considerably bigger. So I don't know what was in the fat cells in the F1. We're just measuring the size. The size was bigger even though the weight wasn't more. So I don't know what was in those cells. What happens in the mesenchymal stem cells? I'm going to show you some genes, but I'll walk you through it. So this gene, zinc finger protein 423, regulates PPR gamma. So you can see that it's up in all 3 generations. Here's PPR gamma, the master regulator of fat cell development. It's up in all 3 generations at all 3 doses of tributyl. PPR gamma regulates fatty acid binding protein 4, lipoprotein lipase, the most important cell in fat metabolism. That gene transfers fat into your fat cells. It's up in all 3 generations, but look in the F3 generation. It's off the charts. We have to break the scale. And that's very different from a genetic effect. The genetic effect is diluted over time. So if you cause a mutation in 1 generation, and then you mate that to a wild type animal without a mutation, the effect is diluted over time. An epigenetic effect gets stronger over time. So that's exactly what we see here. This other gene, PREF1, is an inhibitor of fat cell development, so you can see that it's down in all 3 generations. So we caused a really strong effect here. Because these changes in gene expression were in cells that we just took from the animals, that suggested to us that we probably had an epigenetic mechanism at play. And I won't go through all the reasons why we think that. So we decided to do what's called an epigenomic analysis of the fate of these cells. And we wasted a lot of time and money, I can tell you, studying this with an old-fashioned technique at the University of Arizona. And I was at a meeting and I met a guy named Toshi Shoda, and I was complaining to him about this. And he said, oh, you're doing it all the wrong way. You need to do it by sequencing. And you guys have a very fabulous deep sequencing center here. Toshi said, send Toshi the samples and I will see what we can find. So he did look at the methylation of these, and he could see a certain number of methylation peaks. And he found that 900 were stronger in TBT, the vehicle versus TBT, and about 6,000 were weaker. What does that mean? It means that in the F3 generation, DNA methylation was changed. DNA methylation is one of the classic epigenetic effects that there is. So it was different. So that was enough to let us show that we could get epigenetic changes in the DNA of animals from the F3 generation. And that got us a big grant from NIH to study this in detail. Okay, so we're about a year and a half into that grant. And what we're doing is we're looking at the whole genome. And we're asking the question, what do we see? So F1 through F4. So we went one more generation. Because the director of NIEHS said, well, how do you know it continues after F3? And I said, well, okay, give us more money, Linda. We'll look at F4. So she did. So we're looking at what RNAs are expressed in these cells. We're looking at methylation. We're looking at histones. So the objective is to link transcription, gene expression, with changes in DNA and histone methylation. The idea is to find what did we change in these animals that caused them to become obese. So here's the experimental design again, because we had to do many more animals. We did fewer doses. We only did 2 doses of TBT. And this is the endpoints we looked at, and we went all the way out to F4. And I have to tell you, when we did this experiment, the effect was much smaller than we saw before. It was still significant. And I said to my postdoc, Raquel Chamorro-Garcia, I said, I don't like this experiment. You need to repeat it. This is a year-long experiment. So she was very unhappy with me, but she did it. And the experiment's ongoing now. But she did something very interesting. She did a diet test. So we had some leftover F4 animals that she didn't kill. And she asked the question, what happens if I change the diet of these animals? So they were born, and for the first 19 weeks of their life, they lived on normal mouse chow, which is 4.5% fat or 13% fat by calories. That's a low-fat diet, right? And at week 19, she switched him to a 9% fat diet. Still very low fat, right? And you get 21% calories from fat. And then after 6 weeks, she switched him back again. So what did she find? So this is more data. So you can see here's body weight. So body weight didn't change very much at all, right? It's pretty stable. Here's the lean mass, and here's fat mass. Fat went to the moon. So if I normalize fat mass to body weight, so you can see the percent of fat versus non-fat, look what happens to the lean mass. The lean mass goes way down, and the fat mass goes way up. But just by changing the diet to a slightly higher fat diet, not even a McDonald's diet, just a little bit higher, these animals got very, very fat. And when we put them back to the normal diet, they stayed fat. They didn't lose the weight again. So TBT changed these animals in a profound way to make them respond to diet very differently. Females didn't behave exactly the same. So in females, we can see the body weight stayed about the same, and the fat weight went up a little bit, but that's not what we call statistically significant. So this could have happened by chance. Probably didn't. It means we need more animals in the group. That this number of animals was too small to detect this small of a change in a statistically significant way. Another thing Raquel found is she looked at the livers. I don't know if this really projects well. She said, Bruce, the livers are white. What does that mean? And I laughed and said, that means you get to cut them. So she did that. So she sectioned all these livers and stained them with a dye that stains nuclei called hematoxylin and a dye called oil red O that stains fat. So that's oil red O, it's red. So you can see that everywhere there's fat, is red. So in the vehicle with the rosiglitazone, no fat. In the TBT, at all 3 doses and all 3 generations, these animals had fat in the liver on a normal diet. Another thing we have besides an epidemic of obesity is an epidemic of fatty liver, especially in children. And fatty liver is a health risk for a variety of conditions. Interestingly, the females looked almost the same, except that rosiglitazone caused females to accumulate fat in the liver. So there's a sexual dimorphism. So there's a difference between males and females in how they respond to rosiglitazone, but not to TBT. So TBT causes these animals to have fatty liver. So let's look at some more genes. So these 3 genes are involved in the synthesis of fat. And you can see that they're up. Here's PPR gamma, up in all 3 generations. This one, this one. These genes are involved in burning fat, fatty acid oxidation, PPR alpha, acyl-CoA oxidase. These are all up. So the liver is making more fat, and it's trying to burn the fat to compensate, unsuccessfully. This gene, FSP27, makes the coating of the balls of fat in the cell. So we're making more fat. So what did I tell you about obesogens and development and health? First thing is that I didn't show you all these data, but obesogens are very potent activators of these 2 receptors at the kinds of levels at which we're exposed, at environmentally relevant levels, parts per billion. Tributyl drives fat cell differentiation in cell culture. Exposure during development induces fat in the 2 species that we work with, mouse and frogs. Other people have worked with zebrafish and medaka and fathead minnows and even pygmy sperm whales. TBT causes fat development in all of these. The effects of maternal prenatal exposure are transgenerational. They go all the way out to the F4 generation. TBT exposure alters the response to diet composition. So when we change the composition of the diet, the animals get fat really fast. They're also resistant to weight loss from fasting. I didn't show you those data, but if you fast the animals, which we do before we harvest the tissues, the vehicle-treated animals lose weight really fast, and the TBT animals don't. That's every dieter's nightmare, right? So you starve, you lower your caloric input, and you don't lose weight, whereas other people do. That's exactly the same thing happens to our mice. There's multiple ways that tributyltin can work. I've shown you data on this pathway, but it can also affect aromatase expression that affects estradiol, and it can affect the levels of stress hormones and the mitochondria. So we don't know yet how all these factors interplay, but we know that tributyltin causes this effect. So people always say, well,